Okay, good morning or good afternoon, wherever you are joining us from today. My name is Adam, and I am a webinar representative on the Trainee Executive Committee this year. And it's my pleasure to introduce our February edition of the webinar series, Professor Darrell Mousseau. Darrell received his PhD in neurochemistry from the University of Alberta and did postdoctoral training at the University of Minnesota and at the University of Montreal. He is now a professor in the Department of Psychiatry and the director of the Cell Signaling Laboratory at the University of Saskatchewan. Until recently, he was the Saskatchewan Research Chair in Alzheimer's disease and related dementias, funded jointly by the Alzheimer's Society of Saskatchewan and the Saskatchewan Health Research Foundation. His research has revealed that molecules associated with depression are also influencing models of Alzheimer's disease, which can explain how depression increases the risk for developing Alzheimer's disease. His research also found striking differences in these molecules between the male and female brain. He now believes that Alzheimer's disease follows different courses in men and women, and that the disease will likely require different treatment options based on the sex of the patient. More recently, his group established brain organoid technology, also known as brains in a dish. These brain organoids provide a brain-like environment for the study of human brain diseases such as Alzheimer's disease and amyotrophic lateral sclerosis. These brain organoids could also help with confirming a diagnosis in patients who may be at the earliest stages of either disease and could be used to determine which treatments could provide benefit to the patient. The brain organoids provide a cutting-edge training environment for the next generation of neuroscience and organoid researchers. And with that, Darrell, it is all yours.
Thank you so much for the introduction, Adam. I think you've pretty much given my presentation for today, so we'll just take it really easy here. So I've got quite a few slides here. I'm going to give you an overview basically of where I'm coming from and what I mean by the brain organoid, a window into the donor's brain. And you can hold your questions until the end, or if you feel comfortable with it, you can certainly interrupt me as we go along. I'm certainly happy to do that and answer any questions or comments you might have. So before I do start, I want to acknowledge that even though we are joining from different areas across Canada here, I personally live and work on Treaty 6 territory and homeland of the Métis, and of course we spend our time and effort to make sure that we understand that and we reaffirm our relationship with one another. And I should probably mention these folks off at the start because they're the ones that made much of the research that goes on in my lab possible, and certainly the brain organoid work that is going to be discussed a little bit in the second half of this presentation today. That comes from a private donor, the Legions Fund, down highlighted below in red. These were local donors who were willing to fund research to look into an alternative to animal-based research. They're not they don't support animal-based. So at the time, it was good timing. We were getting out of animal models for various reasons, and we decided to look into the brain organoid. And I think that I've certainly been pleased with that decision, and hopefully you'll understand why by the end of this presentation. So learning objectives for today, you'll see here, I'll give a very general overview of Alzheimer's disease, as well as models of Alzheimer's disease, a couple of 3 or 4 slides about what brain organoids are and why they represent the brain milieu. And then a couple of results just to show that they are useful tools, not simply make project or make work projects for graduate students and undergrads. Like they actually do inform on a number of different aspects of brain function. And then a slide at the end just discussing possibilities. So Alzheimer's disease, this I'm not sure how many of you actually do research in the Alzheimer field, but it all began with a paper published back in 1907 by this fellow up here right in the middle. And he was discussing one of his patients, Auguste Dieter, who was fully demented when he met her, and he then analyzed her brain postmortem. And this was one of the first instances of the description of what is now referred to as the hallmarks for Alzheimer's disease. He noticed these histopathological changes, which we'll get into a little bit over the next couple of slides. I just wanted to draw your attention to the fact over here on the left. This is a survey of PubMed in November of last year, where to date, there are almost 300,000 papers that have been published with the term Alzheimer either in the title or in the abstract itself, 70,000 of which are actually reviews. And bear in mind that reviews are usually a summary of what's known at the time or the author's personal opinion on what might be going on in terms of mechanism or in terms of challenges or whatever. And all that to say that we still have no idea what's going on with Alzheimer's, which is the problem. A lot of it tends to be repeat research, confirmation research, and folks who tend to stand out here asking questions that might be not quite in line with the current dogma tend to be not as I don't know, not as well supported, let me say. Now, the problem with that, of course, comes from many different sources, not the least of which is the reviewer that might be reviewing grant applications or papers. They say they want you to think outside of the box, but then they tell you, well, there's another box you should really think in. They're not quite comfortable stepping outside of that comfort zone, outside of the box. So, what do we know about Alzheimer's disease? Well, it's progressive. It takes, depending on the sources, depending on the reports, it can be anywhere between 10 and 20, 25 years for it to progress to the point where it presents with symptoms in the clinic. It's degenerative, it's neurodegenerative, and of course, one of the main features or phenotypes of Alzheimer's in the clinic is dementia, changes in cognitive status, mood disorders as well, a variety of different things, none of which are very pleasant for the patient. It's very frightening to actually start to realize that you're in that state and then develop further. And of course, for the family that's associated with anyone who's had Alzheimer dementia, Alzheimer's disease-related dementia, they realize that it's a very frightening disorder. There are different types of Alzheimer's disease. There's the early onset or the familial, the FAD. Usually the onset of this form of Alzheimer's disease is prior to the age of 60, 55 or 60 years old. And most of these, the familial forms at least, are dependent on mutations in a couple of genes. The primary one would be the presenilin-1 gene that encodes for the presenilin-1 protein. These cases, though, only account for less than 2% of all cases of Alzheimer's, and they're highly penetrant, meaning that if you carry one of the alleles, you're going to get the disease. The predominant form of Alzheimer's disease is referred to as late onset because it occurs in individuals 65 years or older. The risk of developing Alzheimer's at the age of 50 years old is about 1%, and it doubles every 5 years after that, until the age of 85, when your chances of developing Alzheimer's are 1 in 3, which is a horrific statistic. There's no real heritable or genetic association with this form of the disease. The cause is unknown. Hence, it's also referred to sometimes as sporadic Alzheimer's disease. And again, like I said, this is the majority of cases, 95 or so % of all cases. And the different cases there are different risks, let's say, associated with Alzheimer's, this late onset form of Alzheimer's. Some of these things that are listed here increase risk, and some of them decrease risk. And I'll just highlight some of those in the next slide here. And I always make it clear to individuals that I'm lecturing to or presenting to that a risk is that it's just a risk. It doesn't mean you're going to get it. It just means that it's going to push you one step closer to potentially getting it. And if you want to look at risks as a type of, let's say, a stair in a set of stairs, and let's say you need 10 stairs to get to Alzheimer's disease, and each risk might be worth 1 stair or 2, whatever, depending on how high of a risk it is. Again, just because you have one of these risks does not mean you're going to get it. But yet, if you have a number of these and they all add up, then your chances of developing Alzheimer's disease over time are much greater. And you see here some of the things that have been associated with it. And sleep disruption, continued stress, diabetes as a risk factor. Cardiovascular changes have been associated with high risk of Alzheimer's. Cholesterol, obviously, with plasma membrane component, but also for transport mechanisms within the brain and out of the brain. and even mental health status. Again, just because you're depressed doesn't mean you're going to develop Alzheimer's. It's just a risk, but you start to add them all up, and then eventually you could develop the disease. So one of the issues, well, one of several issues with Alzheimer's disease is that it's a disease of exclusion. And basically what that means is that it's only ever suspected. And that's based on the review of the symptoms that present when you go see the clinician for the first time, the geriatric, the person with this, the specialty clinician. They basically can go through a list of different types of dementias, and they say you don't have this type or that type or this type or that type, and they basically exclude all the other ones and say it's very, very likely you have Alzheimer dementia. But the only way that you can confirm this is on autopsy, when they look again for these histopathological signs that um, Alzheimer's mentioned in that paper way back in 1907. People are still looking for amyloid plaques and neurofibrillary tangles. And I'll go into that a little bit in the next slide just to give you an idea of what those mean. But you can see where the challenge is here is that if you don't know what's actually causing Alzheimer's disease, the challenge is, well, how do you diagnose it in a timely manner um, without having to wait until someone presents with such advanced symptoms that it's almost it is irreversible, but it's almost impossible to manage. And therefore, also, it's a challenge for developing a cure. So diagnosis is an issue, and developing a cure is an issue. When they talk about these two histopathological markers, they're talking about the beta amyloid plaque that's up in the upper left here, and the neurofibrillary tangle. And the beta amyloid plaque is just an accumulation of these aggregated proteins, very hydrophobic, neurotoxic as well. And they tend to aggregate in the form of these plaques, which can be identified with a number of different stains, antibodies, et cetera, et cetera. The neurofibrillary tangle is basically a dystrophic neuron. You can see here that once the axon of the neuron starts to lose its integrity, it starts to get a little bit of a corkscrew shape to it. And these are very, very characteristic of the Alzheimer brain. Here I've got a little cartoon of what it might look like here. Here you've got a healthy neuron and the axon here. You can see here the different proteins, the tau and the microtubule that form the axonal cytoskeleton. They're intact, everything is fine. But what happens in the Alzheimer's state is that you get a phosphorylation of protein tau. And that tau protein, when it's phosphorylated, it starts to dissociate from the tubulins that make up the microtubule. And once you start dissociating there, then you get breakup of the microtubule and whatnot. The plaque, you can kind of see it just accumulating here. And I like to explain it in this way here. If you look at the railroad tracks as being a connection between cell A down here in the lower left and cell B over here, you can imagine the amyloid plaque being like a landslide, and it kind of blocks transport down the axon. And of course, you can see how that would be problematic, not only for um, uh, um, interruption of the axon of the action potential, but also transport of all sorts of nutrients and whatnot up and down the axon to the synapse where they're needed. The phosphorylation of tau, you can see that the backbone is still there a little bit, or the railroad tracks are still there, but the support mechanism is now gone. In both cases, the issue is you're not going to get connection between point A and point B, or point A and point B. They both, however, lead to the same problem, which is a lack of communication, a loss of communication, and the Alzheimer-like phenotype. So the progression of Alzheimer's disease and some of the biomarkers that have been associated with the disease, they're now there have been quite a few of them that have been identified. And as I said before, the disease process can begin 10 to 20 years before there's ever any sign of any kind of a cognitive change, any kind of a dementia state. The problem is that because in the past it was almost impossible to identify individuals who might be on that trajectory to dementia. Here things were absent. There might be a little bit of mild cognitive impairment, this MCI state. but it wasn't until full-blown dementia that people realized there was a problem. You can see how recruiting to clinical trials would be difficult if you had to wait till this stage. You can see how it would be difficult if you wanted to start treating people at this stage because they'd already had 10, 15, 20 years of brain changes. And it would be a little bit naive to just think you could go in there with a drug or an antibody and change the whole brain back to what it was 15 or 20 years earlier. So people have been slowly moving back earlier on in this progression, identifying more and more of the biomarkers, more and more of the states that can be identified that would identify an individual who might be on that trajectory. And the thought is now, if we can identify individuals out here, then we can start treating them out here, and we can avoid all these changes in beta amyloid, which is that plaque, or changes in the phosphorylation of tau, which is the neurofibrillary tangle, both of which contribute to the symptomatology and phenotype. So that's where people are now targeting, is out at this end of the spectrum. Again, with the hope that they can delay this. This is always a little bit amusing, if I can use that term. Many grant applications and journal articles, and this will be a cure. This will lead to a cure for Alzheimer's disease, but it's a little bit naive to assume that a one-size-fits-all approach to understanding the basics of Alzheimer's, let alone to understanding diagnosis and/or how to manage it. This is a little bit naive, and it certainly will probably not be the best strategy to undertake. And I think that over the next couple of slides, you'll tend to agree with me that we've learned a lot, but we haven't learned as much as what we had hoped for. One of the issues, as I mentioned previously, is that there are different types of Alzheimer's disease. So the one in red here is the genetic form. That's the Mendelian early onset Alzheimer's disease. You can see there are other cases of early onset Alzheimer's disease that are not Mendelian. They're non-Mendelian, but they still occur prior to the age of 60, 65. The rest of them highlighted here in blue is the late onset forms. And so you can see it here clearly. That's most of them are the late onset form. No knowledge of what causes it. Certainly no understanding of how to control or treat it. At least not with the one size fits all approach. One of the problems with Alzheimer's disease is that up until recently, it was kind of lumped into one category. It was just Alzheimer's disease, and there were a couple of papers that were published within a year of each other. One of them based on RNA-seq, one of them based on CSF proteomics, and one of them based on imaging, so MRI and PET. And they were able to show that there are probably about 5 different types of Alzheimer's disease. Some of them might be primarily tau. That would be the neurofibrillary tangle, the loss of the cytoskeleton. Some of them might be primarily amyloid. That would be the plaque, the beta amyloid. Each of them having differences in terms of there are different families of genes and proteins that would be up or down regulated. Some of them were not either tau or beta amyloid. It was just immune related pathways. So you can see it would be difficult to try to find a diagnosis that would capture all of these all at once. And that's probably one of the reasons why we just simply do not have as much information as what we'd like. We understand a lot about it. We still haven't just figured out how to take it and translate it to the clinical context. So that was kind of the background on Alzheimer's. I don't want to leave you with that negative feeling. Like, we do understand quite a bit about it. It's simply not enough where we can actually go in with a treatment modality to help the patient. I'll give you a brief overview of some of the models of Alzheimer's, and then, like I said, we'll skip into brain organoids. So there are many different types of models of Alzheimer's. Here I've got some little cartoons. This is cell culture, mice, C. elegans, and even the zebrafish. And there are other models as well. Obviously, there's Drosophila that's out there. People use yeast. People use a variety of different model mechanisms and platforms to answer some of the questions. But basically, the question is, how can we go from a healthy condition to as you're younger, and how that changes and why it changes as you age, and then how does that affect brain function? And these models allow us to test questions on genetics, pharmacology, the response to drugs, various systems that might be implicated using some sort of a pharmacological approach. And obviously, one of the basic questions is, are there sex differences? That's very important because we know that the incidence of Alzheimer's differs depending on the sex. And then, of course, once we've answered questions in these different model systems, then the question is, can we go back to the human brain, and show that there's some sort of a homology between what was found in the animal model or the in vitro model and what is found in the Alzheimer brain? Is there something there that is meaningful and that can be translated? Now, this is a very important paper in the field of Alzheimer's disease. It was published by Glenner and Wong back in 1984, I think, 1984. And this was the first indication of what might be involved, what implicating a specific pathway in the Alzheimer uh, pathology. And basically, what they were looking at was they were looking at the brains of individuals who had Down syndrome, who had trisomy 21. And they found that the individuals with trisomy 21 had a pathology that was very, very remarkably similar to Alzheimer's disease. So they had plaque, they had quite a bit of neurofibrillary tangle, but certainly the plaque. And what they realized was that with that triplication of chromosome 21, one of the genes on that chromosome is a gene called APP, or now referred to as amyloid precursor protein. And they realized that the pathology they were looking at, those little plaques, were composed of a very hydrophobic protein that was a product of the APP gene. So that started the whole study of the beta amyloid hypothesis of Alzheimer's disease. And it's a critical and a pivotal and a seminal paper in terms of um, uh, the Alzheimer field. But I also love to draw the attention to trainees that we're all kind of led to believe that our work is only going to have impact if it's published in journals, the likes of Neuron, Cell, EMBO, PNAS, Nature, Science, and you come away with the feeling that if you're not publishing in those journals, that you're not having an impact on the field. But I want you to bear in mind that this publication was in BBRC, which has an impact factor of, I think it's about 2.1 or 2.3 or whatever. Certainly not up in the sciences and natures and neurons and whatnot level of impact factor. But by far, the impact of this work far outweighs much of the work that followed on with this. So again, impact is not impact factor. An impact factor is just representative of the paper. The impact is it's representative of how your work is going to change the field. Perfect example, BBRC, and this led to pretty much, you know, 30 years of Alzheimer research, and still ongoing. So when we talk about APP, that gene that codes for the APP protein, and beta amyloid, this is what is meant. So APP is it's a large transmembrane protein, type 1 protein, and it can undergo several different cleavage mechanisms. If you look here on the right, it can be cleaved first by an enzyme called alpha secretase, ADAM10 in the brain. And what it does is it cleaves the APP precursor right in the middle of this red rectangle. And you end up with different fragments, and that's fine. And this is known as the non-amyloidogenic pathway. And it'll become clear why it's the non-amyloidogenic when I explain that in the Alzheimer brain, what tends to happen is that there tends to be a shift in cleavage from the alpha to the beta. And you can see here where the two sites are relative to each other. And with beta secretase happening first, and then followed up again by kind of a gamma secretase step, just like it did in this pathway, you end up with the release of this little red rectangle here, which would be beta amyloid. And it can differ in length from 37 to 46, 47, 48 different amino acids. The one that is important in the Alzheimer field is the A beta 42. 42 amino acids, because that's the one that is the primary form of beta amyloid that is found in the amyloid plaque. Hence, then again, the amyloidogenic pathway. Now, just bear in mind, we all have beta amyloids. It's not like beta amyloids don't occur unless it's the Alzheimer brain. That is not the case. We all have beta amyloids of different types. There's a beta amyloid 40, which is the physiological one, which is the primary type of beta amyloid that's formed within the brain. And it far outweighs in terms of amount the A beta 42, which is associated with Alzheimer's disease. But in Alzheimer's disease, the shift seems to increase more the 42 and versus the 40. And again, those extra two amino acids are very hydrophobic, and they lead to a completely different to a peptide with a completely different physical and chemical properties. It's much more. It tends to bind to proteins that it otherwise wouldn't. It self-aggregates. It just messes things up. So again, we all have it. It's just that we tend to have more of the 40 mer instead of the 42 mer. So coming back to models, why did they choose the mouse? The mouse, I'm sure you know, is a great model for a number of different fields of study. In the Alzheimer field, it worked out incredibly well, or I guess poorly for the mouse if you're a mouse. Um, if you look at the sequence of beta amyloid, so that's that A beta 40 that comprises those beta amyloid plaques. A lot of different species have exactly the same, like the identity is 100%, except for the mouse. The zebrafish is completely different, but the mouse is very, very similar to all these other species except for these three amino acids, which means that you could generate a transgenic mouse with humanized beta amyloid and then raise an antibody against the human beta amyloid that would differentiate from the mouse beta amyloid. And this was one of the reasons that the mouse was used. Not the, it wasn't the only reason, obviously. I mean, there's cost and there's the ability to breed and you'll crossbreed and all sorts of things. But this was certainly a major driver, the difference in these three amino acids. So the mouse ended up being the model du jour, so to speak, for Alzheimer research. The problem is the mouse has given us 25, 30 years of results, but none of the treatments that have been designed in the lab have been translated to the clinic. So you can treat a mouse using immunotherapy, using behavioral therapy, using exercise, using diet, using drugs, using this, using that, but very few, if any, of those things translate through to the clinical population. Um, and that's obviously frustrating, I mean, in terms of time and effort, but it also leaves us with big questions as to how to do it, how to treat the disease. Here's some papers that have been published to show that if you're looking at even a particular type of Alzheimer's mouse, so there's the 5 times FAD mouse, which is touted to be one of the best mice to use to study Alzheimer's disease. It carries 5 familial mutations, which of course you'd never find in nature, but this brings all the phenotypes together within one mouse. You get different phenotypes depending on the genetic background that those 5 mutations are backbred into, and this has been shown repeatedly. And I think this one here they actually compared. Um, Let me think. 28 different backgrounds in this report, and depending on the background that your gene is expressed in, you get completely different things and different phenotypes. And in actual fact, in some of the backgrounds, the Alzheimer-related genes actually improve memory and improve cognition. So again, the mouse is great, but you have to understand it's got limitations. And of course, like I said, this has led to quite a bit of disappointment in clinical trials. And here, this is a slide I stole from Tyler, postdoc. Just different headlines showing that, clinical trials basically all sell hope, but they never ever make it to past phase 2. This is one of the first trials that was done with Alzheimer's disease. It was the AN 1792 trial in 2002. It had to be discontinued because the patients who received the antibody, this was an anti-beta amyloid antibody, developed meningeal responses. So basically, inflammation of neurons, um, microbleeds, so microhemorrhaging, which is it just increases risk of vascular dementias and whatnot, and all sorts of other phenotypes that were certainly not helping the patient at all, was actually making the condition worse. More recently, lecanemab was approved by the FDA it was fast-tracked. Again, it was fast-tracked because we're running out of options, to be honest. That's one of the reasons. And people are looking for any kind of an approach to block Alzheimer's disease. And they showed that lecanemab was better for blocking the disease in individuals who were at earlier stages. So again, there was some benefit to that. However, if you went into the supplementary data, which people tend not to read as often as maybe what they should, you could see here clearly that there was a discrepancies between the male and female who might respond to this drug. Ages, clearly, if you're less than 65, it's great. If you're older than 65, it's not so great. If you're white, it's great. If you're black or Hispanic, it's not so great. So again, it's a great treatment, but it certainly has its limitations. And again, recall, like there are different types of Alzheimer's, so the one drug is not going to necessarily fix all. And I do just want to bring your attention to this as well. We tend to focus on these beta amyloid deposits, neurofibrillary tangles, as histopathological markers of the disease. But there are many different things that are going on that could lead to any change in beta amyloid formation or in the phosphorylation of tau. We know there's activation of apoptotic or pro-death signaling cascades. Energy metabolism is certainly affected. There's mitochondrial and ER stress and oxidative stress. We know neuroinflammation is very, very important in DNA damage, obviously, and this is just naming a few. So there are many, many different potential mechanisms at play, all of which could lead to an Alzheimer-like phenotype. One of the problems as well with Alzheimer treatments or treatment management was, although people understood that beta amyloids, so here we have position 1 and position 42, this is your beta amyloid here, the one that forms the plaques. There are different what are called length variants. So there's some that are, they start at position -3 and go to 40 or whatever it is, but there are some that are shorter. They get N-terminally truncated, not just C-terminally truncated. So there's different length variants of the beta amyloid peptide. However, the thought was a beta amyloid is a beta amyloid is a beta amyloid is a beta amyloid. That means they're all the same, when in actual fact they're not. And this is a paper we published a couple of years ago to show that actually some of the shorter beta amyloid peptides actually are neuroprotective against the Alzheimer-related A-beta 42. And we showed this using a variety of different assays for physical interactions, conformational changes using DLS and atomic force microscopy, et cetera, neurotoxicity, changes in behavior, changes in cell viability. And these interactions between these two peptides get even more complicated if you throw a beta 42 into the mix, which is, excuse me, 2 seconds, which just goes to show that in the normal brain, where there are many, many of these beta amyloid peptides, it's a very, very complicated interaction between peptides. They don't just float around and do things on their own, independent of each other. They actually interact, and each one of the interaction could lead to a different cellular and systemic phenotype. And to add to the debate, if I can use that term loosely, there was a study done in 2016 where, and for those trainees that are sitting in on this today, you'd be thankful that you're not part of this lab, because the amount of work that went into this paper was phenomenal. But they looked at 138 mutations of the presenilin 1 gene that have been associated with Alzheimer's, and they looked at 2 different platforms. So they had to subclone these 138 different mutations. Not only did they have to make the mutations and subclone them, but they also just subclone them in two different expression plasmids because they looked at two different assays to confirm their work. And they basically showed that although the understanding or the assumption is that a presenilin-1 mutation that leads to Alzheimer's disease will change that A beta 42 to A beta 40 ratio towards the amyloidogenic type. In actual fact, that's not the case. There were some that certainly did that. Most of them didn't. Most of them, if you looked at the amount of A beta 42 and 40, like it didn't change. In fact, it might even be less than background being the wild type over here, the dotted line. So you can see that it's all over the place with these mutations. And there's now a thought that maybe it's not beta amyloid so much that is involved, but it might be a change in that presenilin 1 cascade that is more important to the Alzheimer phenotype. But anyway, that's not for today. So I the models have been very, very valuable. They've certainly provided inordinate amounts of information, not the least of which would be about the APP and the APP and presenilin-1 mutations, but also the function of the wild types when they compare them. But it's using the mouse and the different models. It's exposed different cell type specific phenotypes, secretase functions, some drugs or classes of drugs that would be beneficial to potentially block some of these cascades. And of course, more in population studies to how lifestyle can affect risk, mental health, and some of the health disparities and inequities that we find within society. And the important thing is because of all the information that we garnered from the mouse and from, cell culture and from whatever, C. elegans, etc., that also all that information is there for us to use so that in case we do go on to develop another model, we can base some of that model development on what's already been gleaned from the mouse. So that kind of segues into the next objective, brain organoids, and just what exactly are brain organoids. Um, so the brain organoid is a three-dimensional culture. It was Lancaster in 2013 who developed this, and the understanding is that the human brain is the most ideal model, but obviously it's just impossible to get human brain samples all the time. So Lancaster developed this protocol where they could take stem cell derived brain cells, grow them in a culture dish, and these brain cells, neurons, astrocytes, and oligodendrocytes, would grow together, distribute, layer themselves, interact in a manner very similar to the brain. But some of these protocols lack the mitochondria, the microglia, and that's a very important brain cell type. It's the brain cells or the brain's immune cell. Um, Up until recently, we're one of few groups in the world that could generate these human organoids in which the microglia do arise naturally. We don't have to add them to the culture from other sources. So that gave us a little bit of an advantage. What does it mean when we're talking about brain organoids? Well, this is it just basically in a nutshell. You collect blood from a donor. You isolate these progenitor cells. You create these pluripotent stem cells from those progenitor cells. And because these are derived from adult sources, you don't have to, there's none of the ethical challenges that are encountered when you use stem cells from embryonic sources. And then you can trick these stem cells into becoming brain cells, and you don't separate them, you just let them grow together. And by virtue of growing together and interacting, they start to connect the way that they normally would within the brain. And it's a wonderful, fantastic model. This is what they look like. You can see some of the layering here, the different markers that we're looking at, GFAP, OLEG2, MBP, and whatnot. And these are you can see here distribution of nu N, which is a marker for neurons, and counterstained with DAPI. So you can see they're a wonderful model. It's not just cells that kind of grow and clump together in some kind of random nonsensical fashion. Like they actually interact in a very, very concerted and organized way. So I'll leave it at that. I'm certainly willing to answer more questions, but again, in the interest of time, we'll jump right into some of the results. And we published this paper recently where we compared brain organoids that have microglia which grow naturally within them, so they arise innately, versus brain organoids in which microglia needed to be added from co-culture. And what we find is that the ones where the microglia are there naturally, they're very healthy. The brain organoids are healthy. It's a great model of the healthy brain. And you can test those healthy brains to see what could lead to a pathology and what not, but they're also very good controls for pathological brain organoids. Um, Here's just some of the results we showed that if you take some brain organoids in which um, microglia grow naturally in them versus brain organoids which you have to co-culture and add microglia to the brain organoid after the fact, if you treat them with different types of beta amyloids, you can see here that the brain organoids take up the beta amyloid. That's not a problem. They'll take it up there. The transport mechanisms are all there. But the brain organoids with the unhealthy microglia, the co-cultured microglia, give an unhealthy beta amyloid profile. This is more like an Alzheimer-like profile. However, the healthy the brain organoids in which microglia grow naturally, you can see that they handle they handle beta amyloid quite readily. This maintains it at the monomer. It deals with it, chops it up, it does whatever it's supposed to do. So that's a healthy brain. But as soon as your microglia are unhealthy, you start to get the banding process here. And that's kind of aggregation, dimerization, oligomerization, etc. So it is quite a good model to study beta amyloid changes. But keep in mind, you've got a nice model of the healthy brain as well. What can we do with this? Well, we did brain organoids. We raised brain organoids from twins. We culture them for 90 days, and we collect um, samples every 10 days in culture. We extract the protein, run them on Western blot, and then we probe them for various markers. Here you can see NUN, a marker of neurons, TMEM, a marker of microglia, synapsin 1, a marker of proteins at the synapse of functionality, GFAP astrocytes, oligodendrocytes, and loading control actin. I want to draw your control here, or your attention to the synapsin 1 in the male. There's a nice expression of it, but in the female, you get this nice glycosylation, this high molecular weight synapsin 1, which is most likely glycosylated. This is very active synapsin 1. So comparing the male and the female already shows, and these are twins, so the genetic background is reasonable. It's not just a random male and a random female and same age, etc. The female is different from the male, suggesting again your connectivity is different, that based simply on sex. Now, more needs to be done rather than just N of one for this, but certainly very, very promising. What about an Alzheimer's disease? Sporadic. I see I'm kind of coming down to 1 or 2 minutes. I think I should be able to get it. The brain organoids from a sporadic Alzheimer donor. Remember I said late onset form is almost impossible to diagnose. You can diagnose the early onset because usually it's genetic and you screen for the gene mutation. But the late onset, it's almost impossible to diagnose. We reused some of the um, the Westerns from the female here, and we compared it to a female donor with Alzheimer's, sporadic Alzheimer's. The cells were the tissue, sorry, the iPSCs were collected before before death, and she was confirmed after death as having Alzheimer's. You can see here the phosphorylation on tau is significant. There's also this cleavage product that we think is a indication of a calpain dependent mechanism. Activation of microglia here a little bit earlier than you see in the controls, but it's higher level. Olig-2 doesn't change, so this is oligodendrocytes, although there might be a loss in the later stages um, of the brain organoids. GFAP, again, not so much of a difference. But look what happens to synapsin 1 in the control. There's that high molecular weight glycosylated form that suggests healthy synapses. There's synapsin one in the Alzheimer one. There's quite a lot of it, but very little of it at that glycosylated stage up here, suggesting that maybe the protein is there, it's just not being properly processed so that it can help with neurotransmitter release and synaptic integrity. And here's a beta amyloid plaque in this same donor's brain organoid. Beautiful plaque. This is beta amyloid, the the same you would see in the human brain postmortem. And just a couple last examples of changes in glycosylation. Here, if you take brain organoids and treat them with the different beta amyloids or combination thereof, you can see that A beta 38, that is, it's a protective one, but it doesn't have to protect against anything if there's no problem. A beta 40 changes the glycosylation state of TMEM119, which is a marker of microglial activation. The A beta 42 gives a different pattern, but now in combination with either A beta 40 or 42, A beta 38 now kicks in and starts to glycosylate this, activating it and making it more capable of uptake of cellular debris. Someone published something very similar to this. It's not us, unfortunately, but it certainly confirms this work. And that was Joe. And Sarah showed that if you treat with a clinically relevant SSRI and antidepressant, you get changes in this high molecular weight, most likely glycosylated MDR1 or P-glycoprotein. So again, can be used for a variety of different mechanisms. And one last result here is ALS, sporadic ALS. Amyotrophic lateral sclerosis, a motor neuron disease. It has a cognitive phenotype. It's very it's a horrible disease. There are familial forms, so genetic forms, but mostly are sporadic. And prognosis with a diagnosis of the life expectancy with the diagnosis of ALS is 1 to 2 years. So it's progressive, but a very, very rapid. But look at here when we compare ALS to Alzheimer's or control, you get neurofilament light cleavage. Phospho-tau, maybe there's some difference, but nothing to write home about. You get cleavage of TDP-43, which is a marker of it used in clinic to confirm sporadic ALS. And you get cleavage also of tubulin, which you don't see in the other conditions. And we thought this might be a loading issue, so we reprobed with actin, and you can see actin is fine. So tubulin's getting cleaved, which means it's really a disease of that neuron. So again, we've used it now to um, uh, diagnose, if I can use that term loosely. We can diagnose sporadic Alzheimer's, sporadic ALS. We can show differences in terms of response to beta amyloids, to antidepressant drugs. We've tried cannabinoids, antipsychotics. It's an incredible system. And what can we do with it? Well, because it's a copy of the donor's brain, we can look for biomarkers that are disease relevant, mechanisms we can study. We can look for cell-specific changes, so microglial specific or GFAP, drug research, like which drugs might be useful if you've got an Alzheimer phenotype. Can you treat and change that phenotype with a particular class of drugs? And then potentially recruitment to clinical trials. Can we do diagnosis? Hopefully screen for those drugs that do change the outcome. So personalized medicine, a variety of different things. And I think I've overdone my time, and I will leave it there. So thank you very much.
That's awesome. Thank you, Professor Mousseau, for that wonderful talk. I did just want to remind our audience to fill out the EDI survey, which should be coming into your inboxes shortly after uh, the webinar. So if the audience could have a look at that, that would be greatly appreciated. I can now open the floor for questions. You can either unmute yourselves or ask questions directly in the chat.
Did I scare everyone? Don't be afraid. Any question's a good question.
Well, I did have a question, so
Sure.
I'll
Go
start
for it, Adam. Yeah.
off. I think in one of your earlier slides, you talked about the evidence of different types of the progression, I think, of Alzheimer's disease.
Yeah.
And I just wanted to ask about your thoughts because you did talk about these immune-related pathways, and if there
Mhm.
was the possibility of designing either monoclonal or polyclonal antibodies at all, and if that could affect any potential treatment pathways for Alzheimer's disease.
I think it's a great question. I think that one of the conclusions that people are coming with is that if you design an antibody that is expected to clear beta amyloid from the brain, it doesn't have as much um, uh, benefit as what you would think it does, it doesn't have as much benefit as what you would hope that it should. So they've shown in many of these studies that they can treat patients with these anti-beta amyloid antibodies. It removes plaque in many cases, but the cognitive deficits don't necessarily change, and the memory certainly doesn't come back. I mean, the brain damage is already done. There have been other antibodies that have been designed to activate microglia. They've been very, very effective because it activates the microglia to clean up the debris. And microglia, I'm not sure how aware you are, but they're basically the resident, immune cell. But they also is the cell that basically goes around the brain and cleans up debris. And they're activated at night when you sleep. There are a variety of different things. So sleep is very good for you. It activates the microglia. They go around cleaning up cell debris. They clean up beta amyloid. They clean up all sorts of things. And there are antibodies out there that have been shown to activate those microglia specifically, which is great. You start to get clearing and degradation again the brain's garbage. They've shown that very, very nicely in mice. It has to progress through to the human yet, but there are definitely going to be antibody strategies that are very, very useful, and certainly targeting the immune system, I think, is going to be the way to go. We've kind of been pigeonholed, or we've had the blinders on looking at beta amyloid for so long that even if you come up with a great story about the immune cell, it has to involve beta amyloid. If you're looking at astrocytes, well, how does it affect beta amyloid? It's like, well, maybe it doesn't affect beta amyloid. Maybe it's doing something completely different. Don't let your bias come in and try to ram beta amyloid into everything. It might be that beta amyloid changes is a marker of brain change and nothing more than that. And the more brain change there is, the more beta amyloid there is, and therefore the more plaques. Okay, but it doesn't necessarily mean that that's the end all be all and the target. So certainly immune cells, antibodies will work, but maybe we kind of think outside of the box a little bit, and we'd probably get somewhere. Yeah,
Okay, thank you, thank you. We did have a few questions come in from the chat,
perfect.
so I'll just sort of go in order.
Sure.
Hello, very impressive presentation. Have you tested the functions of microglia in the brain organoids you made, and how?
So the only way that we tested the functions of the microglia in our brain organoids has been with that comparison with where we had the brain organoids that grew microglia naturally versus the ones that you grow in co-culture. And the interesting thing about that, just as an aside, it's far cheaper to actually grow brain organoids with microglia in them than it is to grow microglia in isolation, grow brain organoids, and then add the microglia to them. But again, long story short, we've only looked at the comparison in that one paper. We've done some other things where we've looked at TMEM, which is a marker of microglia, and IBA1, which is also a marker of theoretically of activated microglia. The interesting thing is we get a different pattern if we look at those two markers. So TMEM 119 is not necessarily, which is a marker of microglia, is not always the same state as the IBA1, which is interesting, but we haven't figured out where along the lines those two might overlap and where they don't overlap. So again, great question, but that's all we've done to date.
Awesome. And then we had another question. You listed a number of risk factors for Alzheimer's disease. Is there any one of those that stands out based on evidence?
Age and sex. The older you get, your increased your chance of developing Alzheimer's increase. So, like I said, every 5 years. after the age of 50. And there's evidence out there to show that women are more likely to develop Alzheimer's than men. However, the debate is still open on that one because there are some population studies to show that the same number of men and women develop Alzheimer's disease, but that men die sooner. And therefore, women, because they don't die as soon, are the ones that seem to, quote unquote, hang around with the disease. And that seems to bias the stats to say, oh, more women have the disease. But those are the two biggies, age and sex.
And just in the interest of time, maybe we'll field
Sure.
one question. Do you think that the link between depression and Alzheimer's has to do with the commonality of insomnia in people with depression, and thus the plaque removal is reduced?
Great question, because we know that the neurochemical systems that are tied in with depression overlap almost 100% the cognitive systems that are tied in with Alzheimer's disease. So those two systems are literally you can overlap them in the brain, suggesting that there's a very, very tight connection between depression and Alzheimer's. The fact that they both have a phenotype, a sleep symptom, is certainly adding to the disease process. And I think that like sleep is very, very important. It's been shown in mice, it's been shown in humans, so it's not just a mouse thing that if you have a good sleep, you usually wake up better. You don't have that brain fog. And in time, like there was a study that was just published to show that there was a really wonderful clinical response to individuals who take sleep medication, that they actually have lower beta amyloid burden and they actually have better cognition. So again, it's an indirect study that wasn't looking necessarily at beta amyloid burden and cognition, but they noticed this as a side effect of of these sleep medications, that people wake up without that brain fog and beta amyloid burden. So Alzheimer like beta amyloid burden is actually reduced. So again, very, very strong evidence that sleep is very important against Alzheimer's.
Excellent, excellent. So we are just at about the top of the hour, so that
Perfect.
is our time. But I just want to thank Professor Musso again for giving a very insightful talk and for the audience for attending. And I hope that everyone has a wonderful rest of their day.
Yeah, enjoy your reading week, everyone, and thank you very much for joining us today. Cheers.